Influenza is the thing that makes us most worried in public health. Even with the deadliness of COVID, we think a flu pandemic could be even worse. The only thing you can say for certain about flu is that it's unpredictable. That's the only thing we can predict.
A strain of bird flu is sweeping the world. It has caused a pandemic in animals that may well be the worst on record. It's estimated that over 280 million birds and tens of thousands of mammals have died worldwide.
What we are seeing is just the tip of the iceberg. Highly pathogenic avian influenza virus has been detected in over 500 different bird species and also in over 80 different mammal species. So it's not only birds, but it's also wild mammals that are at risk.
This particular strain of bird flu, known as H5N1, keeps on doing the unexpected. And now experts are concerned it could become a human pandemic. After first emerging in Southeast Asia in 1996, it was spread around the world by migrating wild birds. Dr. Lineke Begeman was part of an expedition investigating reports of dead birds in Antarctica, one of the last remaining regions previously untouched by the virus.
We travelled about a year ago in March 2024. So that was right after the first case was detected. Our expedition was aiming to at least cover a certain area that we heard that there were mortalities observed, to see if it was indeed avian influenza or if it was something else.
This pristine environment is home to some of the most well-known animals on Earth. 63 bird species, including the iconic emperor penguin, breed there each year. Large colonies of sea mammals, such as crab-eater seals and elephant seals, also call it home.
We had a team with virologists, wildlife biologists, wildlife veterinarians, and I was a veterinary pathologist to understand the cause of death. And we had this laboratory on board, so we could immediately test and we could immediately adapt our strategy and our travel to our detections. And of course, it's also challenging because it is an environment where you need to think about your own safety. I needed my fingers to continue to do delicate work, so I couldn't wear a lot of gloves, so I actually froze my hands. The skin would come off.
That sounds terrible.
It was terrible. So it was physically challenging as well.
As well as frozen fingers, they also had to contend with frozen animal carcasses. Once they had finally managed to extract samples and take them back to the onboard lab, they found the results were as they feared.
So we managed to visit 10 sites. Out of those 10 sites, we detected highly pathogenic avian influenza in 4. All of these sites and detections were in skua that were found dead. There was a really big outbreak going on with over 50 animals found dead and about 80 animals still alive. What is really interesting is that there are very few, so it's only about, population is about 20 to 30,000. That's it.
The virus is still circulating in Antarctica, and the latest cases were recorded on several islands just 450 kilometres away from Oceania, the last area free of H5N1.
Those islands contain a lot of special bird species. They might well be sensitive for this virus, so there's a big concern that again this virus will cause a high mortality and it puts a lot of pressure in the conservation of these species.
There's little to be done to stop the virus from spreading among wild animals. However, scientists continue to monitor and document confirmed cases using the data to support conservation efforts for the most severely affected species. So why did this H5N1 strain of bird flu become so deadly? Professor Debbie van Riel at the Erasmus Medical Centre in the Netherlands has been studying this virus for the last 20 years. What motivates someone like you to spend most of your career looking at these tiny virus particles and trying to understand them?
So it started off that I'm really curious, understanding how a virus, a really tiny thing, can cause really severe disease, and understanding how this can happen. Some viruses cause mild disease, some viruses cause severe disease, so what triggers this difference? And if there's one thing that we know of influenza virus is that it can cause pandemics.
So this highly pathogenic virus that we're talking about in this film, why is it called H5N1?
Well, I've got here a model of influenza virus.
Well, that was handy.
Yeah, it just happened to be here. And it has 8 gene segments.
Right.
And all of these gene segments encode 1 or 2 proteins, or sometimes more.
Yes.
2 important proteins encoded by these gene segments are surface proteins.
Yes.
Like you can see here, the balls on the outside of the
Right.
virus. And these are either called the hemagglutinin, so the H,
Yes.
or the neuraminidase, the N.
What are their different functions?
So the hemagglutinin, so the H, is important to attach to the host cell, which is really the first step of infection.
Right.
The neuraminidase is important for the release of the newly produced or the newly assembled virus particles from the host cell.
So H lets the virus into the cell, and then it replicates inside the cell, and then N causes the cleavage that releases that virus.
Indeed.
There are 19 known hemagglutinin H and 11 known neuraminidase N subtypes. Any combination of H and N subtypes is possible, which is how we get names like H1N1, H3N2, our seasonal flu, or H5N1. These surface proteins are essential for the virus to infect cells and are the primary targets of the immune system, particularly antibodies. Changes to H and N can enhance the virus's ability to infect and spread to new species. But how do these changes occur? Viruses can't reproduce on their own. After attaching to a host cell, they are absorbed and hijack the cell's genetic machinery to replicate. During this replication process, RNA viruses often introduce errors into their genetic code. These errors actually form part of the virus's evolutionary strategy to evade immune defences. Most of these changes are harmless or even harmful to the virus, but occasionally one gives it a crucial edge. If such a mutation occurs in the wild, the chances are the bird will die before having a chance to pass it on. But when a low pathogenic virus enters a crowded poultry farm where thousands of birds live in close quarters, it can replicate billions of times in a short period. With so many chances to mutate, the risk increases that the virus will evolve into a strain that's more virulent. That's how the H5N1 subtype first emerged in China in 1996. However, this was far from an isolated event. Other highly pathogenic strains of avian influenza have been linked to intensive poultry farming operations across the world. The H5N1 virus was then passed back from farmed birds to wild birds, where it was spread further. At the University of Cambridge, Professor James Wood is studying infection pathways.
It's evolved into a strain that transmits through a really wide range of different species. So you have the opportunity, once migratory birds have brought it into a country, for it to spread through potentially every species of bird there. So that massively increases the range of problems that it can cause. And also therefore makes far more farms likely to be impacted by it, because not every farm will have ducks or gulls on it, but most of them will have things like sparrows or pigeons or other sorts of birds. And once one bird in a flock gets exposed to it, because it's so highly contagious amongst birds like chickens, then it will spread very, very rapidly through a laying flock or a meat flock, and cause really big mortality.
Between 2021 and 2023, an estimated 50 million birds were culled across the UK and Europe, and another 148 million in the United States since 2022, in efforts to contain the spread of the H5N1 virus. Professor Wood is part of a consortium of experts studying the virus in the UK. They found that wild birds are now the primary route of infection into poultry farms, overtaking the movement of infected poultry between farms as the main transmission pathway.
And the area that we've been working on has been looking at what happens on farms to try and understand far better how farmer behaviour can both protect or actually make farms more susceptible to the infection getting into their birds from wild birds.
What sort of behaviours?
The preventive behaviours that we try and promote, loosely termed biosecurity measures, are all about farmers making sure that they're scrupulous with their cleanliness.
How are you finding farmers are reacting to this with regard to compliance?
Many farmers are doing a fantastic job, but actually even the ones that do a really good job sometimes make the odd mistake, or sometimes it's not clear how the infection has got in. But generally speaking, when a farm has gone down with it, there are usually some cracks in their armoury against contamination. And this is entirely new for them, because in the past, all farmers have really had to worry about is infection coming in from other farmed birds, whereas now the challenge is coming from almost from within, because of the fact that wild birds will live around most farmed bird premises because there's food or other material that are quite attractive for them.
Biosecurity, surveillance and monitoring are not only vital for protecting livestock and farmers' livelihoods, but also play a crucial role in reducing the risk of the virus jumping from animals to humans. So you're doing a great job containing this very virulent epidemic in birds, but the real concern is that this could spill over and be a zoonosis like COVID.
That is always a major concern with avian influenza. It's not just that it causes devastation in birds, it's spilling over into other species. And over the last 5 years, we have seen at least 2 humans in the UK alone that have been diagnosed with it. So yes, it's a real concern. It's merciful that with this particular strain, it appears that not too many humans have been infected with it. But that's in stark contrast to what's been seen in North America, where they have a slightly different H5N1 that's managed to infect their cattle, where there have been quite a lot of human cases that have occurred, in particular on people working on dairy farms, where so much of the infection has been spreading.
In early 2024, farmers in Texas started to notice an unexplained drop in milk production accompanied by dead cats on a number of farms. After investigation, and to everyone's surprise, it was found that H5N1 was circulating. Dr. Tom Frieden is the former director of the US Centre for Disease Control and Prevention.
Although the first case was not detected until March of 2024, it's almost certain that the disease was spreading among cows since late 2023. One thing to understand is that in cattle so far, although this strain of flu causes some health problems, it's not like bird flu, which causes severe health problems in poultry. The other big difference is that bird farmers understand the periodic need to cull huge numbers, often millions, of poultry that are very sick to protect the other poultry. That's not what is likely to happen with cattle, where you have a very much more valuable animal and you don't have that severe illness. So it's very different both in how it presents and in what the response needs to be.
How is the outbreak being monitored and contained?
The bottom line is that the outbreak is not being monitored or contained particularly well in the US. We're flying blind. We really don't know how widely it's spread. We don't know to how many farm workers it is spread. We know that it is not likely entering the milk supply or the meat supply, that pasteurization would eliminate it. But control is very difficult. And so far, we're really monitoring its spread.
To date, 1,020 herds in 17 states have been affected. There have been 70 confirmed human cases, with transmission mainly linked to raw milk and milking procedures. Most cases have resulted in only mild illness, but there has been one reported death.
We need to continue to improve protection of farm workers, not only to protect their health, but also because one of the big risks here is that this strain of bird flu will combine with other strains of flu, and a new, even deadlier strain will emerge that's harmful to people and to other animals.
Dr. Frieden is referring to a process called reassortment. This is another mechanism by which flu viruses evolve. Reassortment occurs when an animal or human cell is infected by two different types of influenza viruses at the same time. For example, a human-type flu and an avian flu. During replication, one or more of the 8 RNA gene segments can be mixed up, resulting in the emergence of a new hybrid strain that could be adapted to transmit between humans. The 1957, 1968 and 2009 flu pandemics were all caused by reassortment events. That's why quick responses to outbreaks and strict control measures for infected livestock are critical. They not only help contain the virus's spread, but also play a key role in limiting human exposure. The World Health Organization has even expressed concern over the limited information available on bird flu coming from the US since Donald Trump's recent decision to withdraw from the global health body.
We regret the announcement that the United States intends to withdraw from WHO, and we would welcome the opportunity to engage in constructive dialogue.
The new administration has been very concerned by the increase in egg price and the reduced availability in eggs across most North American states. And so they've been looking at even reducing the amount of controls put on some farms. So it does appear that the direction of travel is to reduce controls rather than to increase them.
And the controls in the cattle industry, in the dairy industry, have been criticised as well, haven't they?
There haven't been any real controls in the cattle industry. And that's been most surprising for many of us who work on infectious animal disease control, because even if you haven't got legislation that would allow a really important, potentially zoonotic infection like this to be controlled, you'd want to very rapidly introduce legislation to allow it to be controlled for human health reasons, let alone the animal health reasons. But that hasn't happened.
I hate to use this overused journalistic phrase. That sounds like a ticking time bomb to me.
I think that many of us are holding our breath, just hoping that the infection can be removed from the dairy cattle industry before a real disaster happens.
How would that happen without any legislation, any control?
So the cattle will develop immunity to this, so they don't get infected then stay infected. They'll typically not stay infected for more than a couple of weeks or so. You can sometimes find bits of the virus in milk from dairy cattle for a few weeks, but the real infectivity is no more than a couple of weeks in the vast majority of cattle. So once they've developed immunity, then you hope that the virus will pass on, and if that immunity is lifelong, then hopefully it will protect them.
Since we spoke to Professor Wood, there have been reports of cattle being reinfected with different strains. It was thought the initial spillover from birds to cattle occurred only once. However, the recent discovery of additional spillover events are changing experts' view on how often these events may occur. At this stage, it's likely too late to fully eradicate the outbreak in dairy cattle, and bird flu may become endemic in livestock populations across the US. Apart from a reassortment event, which could instantly create a transmissible new variant, some mutations during virus replication, as we explained earlier, could also make the virus more transmissible between humans over time. Professor Van Riel's recent study had looked at where H5N1 attaches and replicates in the human respiratory tract.
The respiratory tract is really diverse. It has many different anatomical sites. It starts at the tip of your nose and it ends deep in the lung. Seasonal influenza viruses that cause epidemics every year can attach and infect to cells in the nose and in the trachea. So that's why they can be transmitted so easily. On the contrary, if you have a virus that would need to infect cells deep in your lung, that would be more challenging for the virus to infect someone because it needs to travel all through the lung, and on its way it might be neutralized by mucus or caused away by mucus or immune cells like macrophages, so that's less likely to occur.
Traditionally, avian flu viruses mainly infect cells in the lower respiratory tract. Professor Van Riel and her team have been investigating whether the H5N1 virus has been adapting.
So this is a very thin section of a human trachea, and we incubate it with the virus of interest, and we add a red label to it.
To the virus?
To the virus.
Right.
So this would be the air that you breathe.
Right.
And this is the epithelial lining of the trachea. And here you can see all the tiny cilia. And so the red signal is binding of the virus to the epithelial cell. This is a seasonal H3 and 2 virus, so a virus that is circulating throughout the year and causes yearly epidemics. And you can see that these viruses can bind very efficiently to the cilia of the epithelial cells.
So, just to reiterate, what we're looking at here is the common human flu, H3N2, which circulates around the world every year. So that one's very much at home. It's doing really well.
It does very well. We know it can bind, essential for infection. We also know that it can infect.
Right.
So this is the same tissue section, but then incubated with a H5N1 virus from 2005. And you can see, we do see very occasionally that the virus might bind, but in general, there's very little attachment.
So it's not doing very well at all?
No, and the fact that it doesn't bind fit with the fact that these viruses cannot efficiently replicate in these cells.
Okay. What's next?
Next, this tissue section is incubated with a more recent H5N1 virus.
What year is that one from?
This one is from 2022, and what we see is we see more attachment to these cells.
So this one from 2022 is doing much better than the one from 2005.
Indeed, it suggests that human can become infected more easily with recent H5N1 viruses that are currently circulating.
Does that make this a likely candidate to be the next pandemic we need to worry about? Is it telling us that?
If I would know that answer, I would love to give you a good answer to that. We don't know. We know a few characteristics important for influenza viruses to become the next pandemic. And one of those things is that they should be able to attach and replicate to cells in the nose and in the trachea. The attachment pattern that we see is not yet as good as we would expect from a virus that would cause a pandemic, but there seems to be a change, at least. So it's a sort of intermediate phenotype.
Professor Jim Paulson and a team at Scripps Research in the US have been trying to understand how natural mutations could arise and what their impact might be.
All major human pandemics originated from avian viruses. So what we found was a long time ago is that bird viruses recognize different receptors than the human viruses. In order for the bird virus to recognize the human receptors, they have to mutate in their binding protein. And it's just a few amino acids normally that was required for the virus to switch specificity from avian type receptors to human type receptors so that they can transmit from human to human. But in order for the virus to transmit from human to human, now you're talking about a sneeze with droplets that contain very tiny amounts of virus. So for the virus to successfully latch on, if you will, to us, the cells lining the airway, it has to recognize these receptors in order to efficiently infect. And so that's why this property is important for the human-to-human transmission.
The results from their recent study showed that over the past 10 to 20 years, the H5N1 virus has evolved significantly. It once required 3 or 4 mutations to adapt to human-type receptors, but now it might just need 1.
So there was always some comfort taken that with a number of changes that are needed, that this was the reason the virus wasn't emerging to be able to transmit from human to human. So now what our paper showed was that it just increased the risk of the virus being able to acquire that one property that we know is needed for human to human transmission.
The mutation they found takes place in a single amino acid in the hemagglutinin, the surface protein that attaches to host cells. What's the main message we should take away from your research?
Our main message from that is really that we need to be on high alert in case this does occur, and if we do see human-to-human transmission, then to be prepared for that to prevent a severe pandemic.
One thing we forget when we're making shows like this is that Every year, 650,000 people or thereabouts die from influenza anyway. So why are we worried so much about this one?
First of all, there's no herd immunity, so the whole population is naive. So we know that the first infection often results in more severe disease than subsequent infections. The second thing is that this virus, as it is now, is more virulent than we know from seasonal influenza virus. So we know that it can cause mild disease, but we've also seen cases of very severe disease, not only in humans, but in many mammalian species. So it can, for example, cause a severe pneumonia, it's a respiratory virus, but it also has the capacity to cause a severe encephalitis or even spread to other parts of the body.
The 1918 flu pandemic, caused by a novel or recently evolved influenza virus, is believed to have killed between 50 and 100 million people worldwide. The good news is that today we're far better prepared. A vaccine targeting the H5N1 strain already exists, offering a critical line of defence. Governments around the world are starting to stockpile H5N1 vaccines, with Canada already making 500,000 doses available to farm workers. Vaccinating livestock is also being explored as a preventative measure. Trials in several countries have shown promising results. In China, for instance, a national poultry vaccination campaign targeting a different strain of bird flu significantly curbed the virus's spread and eliminated human infections linked to poultry. The risk of bird flu to the general public is currently considered low, and the situation continues to be closely monitored by health organisations. While pandemic preparedness, such as vaccine development, is underway, there doesn't seem to be a clear-cut answer. Is there a solution? What do we have to do?
We cannot stop this outbreak, but we can observe what species are mostly hit, and we can try to protect those species by making it as easy as possible for those species to restore themselves. So that's the direct solution, and I think the long-term solution would be to prevent such an outbreak in the future, and this needs a massive change in the way we keep livestock. But it will be not only beneficial for wildlife, but it will also be beneficial for our own health and for the health of the poultry.
People will say that it's not if, but when. And I think this is where having really good pandemic preparations is a really important part of our response. But I think as well as those sort of technical measures, really important tried and tested measures which really relate to stopping exposure between animals and humans. And a lot of that will happen at the farm interface.
The only thing you can say for certain about flu is that it's unpredictable. That's the only thing we can predict. Does it mean that a pandemic's coming? No one knows. Does it mean we should improve our preparedness? Absolutely.